Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about gel electrophoresis of the amino acids. And here's the problem: if a mixture containing both glycine and cysteine is buffered at pH 5.5 and then undergoes electrophoresis, show the results of the experiment. And we are told that isoelectric point of the glycine is at pH 6.1 and isoelectric point of cysteine is at pH 5.0. Take a look at this picture, which shows us average amino acid structure, which has amino group, carboxylic group, central alpha carbon, hydrogen, and R group. R group would be unique for each amino acid, but this backbone is going to be the same for each amino acid. And for example, if we are told that isoelectric point for glycine, and in glycine, our group is just going to be another hydrogen here. 6.1, that tells us that at pH 6.1, we are going to find this amino acid in its zwitterion form. So this amino acid would have positive charge and negative charge, and net charge is going to be 0. As for the cysteine, it's going to have the same zwitterion form with positive and negative charge and net charge 0 at pH 5.0. And what's going to happen to these amino acids if they would be placed in low pH or in acidic environment? Then groups that can be proteinized would be proteinized, because in high acid or low pH, we are going to find a lot of free hydrogens or protons. Protons is going to be hydrogen without electron. And this carboxylic acid group is going to be proteinized, and that means that this group is going to lose this negative charge. But if our solution is going to be basic in higher pH, we are going to find a lot of hydroxides which are going to steal this hydrogen and would form a molecule of water. So this carboxylic group is going to lose hydrogen and would get a negative charge. And this amino group is going to lose hydrogen and would be uncharged. So net charge of such molecule is going to be negative. Unlike when we want to separate DNA, we load DNA on just one side. For example, it's going to be this side, and then we apply positive current here and negative current here, and DNA, because being on the overall negatively charged, is going to move to this positive pole, to this positive charge. But with amino acids, the story is different, because amino acid may have both positive charge, negative charge, or can be in zwitterion form where net charge is going to be 0. So we load amino acids here in this region in the middle of our gel. And when we run a gel, molecules of amino acids, depending on their charge, are going to move whether to this negatively charged electrode or positively charged electrode. According to our problem, pH of the buffer is 5.5. What's going to happen with glycine, whose pH 6.1? This is pH at which glycine is going to be in this neutral form, having both positive and negative charge. So again, pH of the buffer is 5.5. So let's say this is going to be 5.5, and glycine is 6.1. So it's going to be here 6.1. So as you see, buffer is more acidic than pH of the glycine at which it's going to be in this zwitterion form. So we can find glycine at 5.5 majority is going to be positively charged. So if glycine is going to be positively charged, if we load it here, it's going to move to this negative electrode being positively charged. Now let's take a look what's going to happen with cysteine, whose isoelectric point is going to be at pH 5.0. So would be somewhere here, 5.0. And as you see, our buffer, pH 5.5, is to the right. And what's going to happen to this molecule, which would be in Zwitter ion form at pH 5.0, if we move to the right. And in this case, this molecule would be predominantly found in this form, which is going to be negatively charged. So if we have molecule that is going to be negatively charged, so negative charge, it's going to move in the direction of the positive electrode. So this is going to be a solution, but probably some of you wonder what's going to happen to amino acid whose isoelectric point is going to be 5.5. So such amino acid at this pH, which is going to be pH of the buffer as well, would be found in the form of Zwitter ion with positive and negative charge here. So net charge is going to be 0, and it's going to stay in the same place and not going to move anywhere. So we will find the band which is going to be here in the middle. So this is how scientists separate amino acid, just changing pH of the buffer and knowing isoelectric point of different amino acids, we can predict in which direction they're going to move. And this is all for today. Subscribe and see you in the next video. Goodbye.